Very good morning, students. Welcome back to the class, Immunology and Immunotechnology. Welcome back to lecture 9. Today I will be talking more about MHC molecule and what are HLA and what are HLA types. HLAs are nothing but MHC molecules only. In humans, they are called by HLAs. We will see why they are called HLAs, and then a little bit about genetic control of immune response. These will be the topics of interest for today's class. So the outline is simplified in 3 concepts. The first concept is organization and inheritance of MHC genes. We will be specifically talking about MHC genes. What are those? Excuse me. And then where they are organized in the chromosomes, in the genome of humans, and then how they are inherited during development. And then structure of MHC molecules, we will talk further. And then central function of MHC molecule in immune response. We will look into that. So please be noted that these some of the lectures I have done with some other additional material available in the internet, SlideShare and other presentation from different authors. So as per, I'm not using it for monetizing purposes. So I'm just using for educational purposes. This is the copyright policy. As long as I don't use to get money from their work, I'm fine to give because I'm sharing the knowledge to my students. Recap in terms of fundamentals. Coming back to fundamentals of immunology, just glance through quickly 1 to 2 minutes on the few slides so that you brush up your memory. So innate and acquired immunity, innate and adaptive immunity, different cells of the immune system, crosstalk between the two fundamental immune pathways that work in. And again, one of the concept of inflammation and phagocytosis we studied. And coming on to specifically one of the type of cells, T lymphocytes, we came across about specific antigen presenting molecules. They bring on their cell surfaces specific molecules called MHCs. Along with MHCs, the antigen will be processed and presented to the T cell. That is the T cell mediated immunity that we covered quite large classes. And then in terms of self and non-self antigen, the self marker will have an MHC molecule that label as, and then non-self will have an antigen that without MHC, and that the distinction happens at the fundamental level, self and non-self. This is an antigen, non-self antigen. This is MHC can be considered as an antigen itself, but it is self antigen. Remember that fundamental difference. And then different organs involved in the immune system, followed by 2 fundamental aspects of immune responses, humoral and cell mediated. Humoral mediated by B cells. When the response comes up, they go into clonal selection process and they divide into plasma cell, memory cells, certain antibodies. Coming on to T cells, we noted the two kind of T cells, T helper cells, T cytotoxic cell. T helper cells nothing but to help the B cells. They are crosstalk with the humoral, and T cell cell mediated, it goes into again a memory cell and active cell, and they use specific mechanism of cytotoxicity. They produce cytotoxins, and specifically fundamentally you see that B cells mainly involved in extracellular pathogens, destruction of them with antibodies, but T cells are in terms of defend against infected cells as whole. In terms of cancers, in terms of transplantation tissues, they are really important. One fundamentally, if you take in terms of the potency of the immune system comparing to passive and active, in the passive and the active again humoral and cell mediated. In cell mediated again T helper T cell, the T cytotoxic cells are the highly potent cells where they can elicit a strong immune response. How the T cells do that? We have already seen that specific antigen processing and presentation mechanism happens. Specific APC antigen presenting cells bring specific molecules of the antigen, and recognition crosstalk with the T cells or cellular interaction, cell-to-cell interaction. And we briefly spoke about some MHC1 protein, MHC2 protein, where they have a distinction between extracellular and intracellular antigens. We will talk further more about these MHCs. And in terms of which MHC is associated with the T cell, which T cell is also fundamentally important. For example, T helper cell, T cytotoxic cell, each of the T cells will have a different association with different MHCs, and how it will be, and we will see in this class today. And remember that CD4 and CD8 molecule, these are the signatures on the T cells, and they will form a ternary complex along with the antigen presenting cell, MHCs. These two along with MHC molecule form a ternary complex. That complex interaction helps in eliciting immune response. That we already covered in fundamentals. And if you take a general metabolic process or general signaling or growth or any of these biological processes that happening in a cell or given cell or an organism, a major proportion or in terms of antigen presentation, MHC molecule, this is constitute high proportion compared to other molecules. But in general growth and transcription, cell signaling takes major proportion. But in terms of immune system, mainly the antigen presentation is giving a major portion of that. So is everything going smooth in terms of audio and video?
Yes, sir.
Okay.
Sir.
So
Yes, sir.
if you look into the structures of the antibodies, T cell and MHC molecule, how they are located in the membranes, as we studied, they are protein in molecular nature. They consist of immunoglobulin, Y-shaped antibody FC and FAB region. T cell receptor, functionally, structurally, it looks like that. All of them will have a common transmembrane domain, either two transmembrane domain, either one transmembrane domain. And you just focus on today's class, MHC molecule. The structure of the MHC molecules looks like that. There are alpha and beta sheets. We will see what are these sheets and how they are associated with the membranes. And that's look into that more details into the class today. But let the outline take into another form in terms of the curricular aspects, what we have studied. In terms of the unit 3, we are already in the unit 3. And unit 3 talks about two concepts, nothing but about MHCs and HLAs. The one concept that they distinguished in terms of structure of MHC molecules and types of HLA system. We will see what are those. And they talk about another concept called genetic control of immune response. So they are talking in terms of what are the genes that control immune response, that called immune response genes. If you take an example of immune response genes, they are operationally defined as antigen-specific genes that control the ability of an animal to raise an immune response, either humoral or cell-mediated, to a particular antigen. They are the genes that will elicit immune response that corresponds to humoral or cellular. We have seen that in humoral cellular, in B cell humoral antibody production, we have seen already that what are the genes that control B cell, how the different variants of the antibodies can be produced with a specific B cell combination. They use a specific fundamental mechanism that we discussed called VDJ recombination. This is somatic recombination and gene rearrangements within zygotic situation. Multiple gene segments are there. They use a specific molecular mechanism called splice variants. And at the same time, when come on to T cells, how the genes, which genes control T cell is nothing but they are indirectly controlled by MHCs. If the genes controlling MHC production, by default, they control specific T cell activation. And what are the genes that control MHCs? Again, there are multiple gene families are there. But the fundamental difference between the molecular mechanism is that in the B cell, there is splice variants, recombination will happen. But in MHCs, production of multiple MHCs, there is no splice variant per se, but there is a specific genetic phenomena called DNA polymorphism. That being said, each gene will have multiple alleles of the same gene in the given locus, and multiple alleles will produce multiple gene products, multiple mRNA molecule that produce a polymorphism. This will be the most of the class we will be talking about it. Then coming on to a little bit more details of what are these MHC molecule, why they are called HLAs, and what is the structure and functions of that. We will quickly go into a few slides. We'll take a little bit slowly from now onwards. So their MHCs are nothing but called as Major HistoCompatibility Complex molecule, MHC molecules. Why they are called by the term histocompatibility? They are talking about in terms of the histo or the tissue, and the compatibility in terms of the tissue given, the tissue of choice for transplantation is compatible with the host and donor or not. In terms of the immunological research, immunological studies in the early 900, most of the studies were concentrated on the transplantations and the anti-sera development, antigen antibody production. So the term refers to the early pioneering studies done on that. So compatible with the host tissue and then the recipient or not. That's why called histocompatible. And then it's a process involved in transplantation. In transplantation, to describe the ability of a donor's tissue or the organ to be accepted by the recipient, and a group of genes that control this process of histocompatibility in all animals is called major histocompatibility complex. Why they are called major? There is another minor group is there. So the major histocompatibility complex group is really important for transplantation in all animals. This is called MHC in general to mice, in general to humans, in general to other organisms. But coming on to humans, they are mainly called as nothing but HLAs, human leukocyte antigens. The MHC molecules are nothing but they are self-antigens presented on the antigen-presenting cells, which are part of recognition with the T cells. They are self-antigen. They are present on the leukocytes, proteins present on surface of white blood cells, leukocytes, and they are specific to humans. That is called as HLA, human leukocyte antigen, to distinguish other species of MHC molecule from human form. You call this major histocompatibility complex in general, and you call it as a human leukocyte antigen coming on to specific to humans. So, in fundamentally, let's talk about different genes that control HLA. We talk about HLA or we talk about MHC. When we talk about structure, we go into MHC molecule. When we talk about polymorphism, we'll talk about HLA types based on the nomenclature. This is because both of them are synonymous. Human leukocyte antigen, human version of the major histocompatibility complex. There are mainly three classes of MHCs, class 1, class 2, and class 3. Class 1 again have a major gene families. There are few other families we can see again. The major gene families are called HLA-A, B, and C. And then class 2 constitute HLA-D. Again, D is divided into 3, DP, DQ, DR. Again, DR is divided into DR2, DR3, and DR4. These are different gene families of class 2. Class 3, nothing but they are specifically some of the complement proteins we discussed in the complement system last class. They also constitute part of the MHC complex and some of the cytokines, but they are very distinct in terms of structure and function from class 1 and class 2 molecules. But we will keep an interest of study for mostly to class 1 and class 2. And the class 1 mainly interacts both class 1 and class 2 as suggested. Their purpose is to help the T cells, that means T lymphocytes. But which class helps which T lymphocytes? Class 1 helps the cytotoxic or tumor suppressor TC cells, they are associated with TC. And class 2 associated with T helper cells, which help in the B lymphocyte recognition process or crosstalk with the B lymphocyte. That means literally class 1 associated with T cytotoxic cells. We already saw that T cytotoxic cells will have a marker of CD8 positive and T helper cells will have CD4 positive. But sometimes it's very confusing to see that one cell, one class, different cells, one class, different cells of the T, again different CD complements. So how do you make them in terms of mnemonics, or how do you memorize in terms of keeping the brain a simple understanding with the neurons? You go with simple example of multiples, how you can produce a numeric 8 by multiplication. You can go with 8 into 1 that is equal to 8, or you can go into 4 into 2 that is equal to 8. In both multiplications, you get a final product that is A. If you remember that 8 ones are 8, 4 twos are 8, remember that mnemonic, you put it into this. 8 is nothing but CD8, 1 is nothing but class 1. So class 1 MHC is associated with CD8 positive, that means T has cytotoxic cells. And then class 2 associate 4 twos are 8, that 4 is a CD4 positive cell, that is T helper cells. They associate with class 2 and the 8 cup. Yeah. This is very beautifully you can remember as mnemonics because you are going into multiple nomenclatures, multiple types in the given process. 8 ones are 8, 4 twos are 8, 1 is associated CD8 class 1, 2 is associated with class 2, 4 is CD4, that gives to the 8. Again, you have to make short of sketches and notes. You go into an MHC in class in this direction, you go with HLA system on the bottom direction, class 1, class 2, and class 3, A, B, C, and D. You can distinguish by different gene families. I'm saying multiple genes still. This one explained from Kube Immunology, chapter 7. But for the present class, I am not following Kube chapter as such, because the chapter has been given a comparative analysis between mice and humans, different nomenclatures. But given the exam point of view, I'm taking only human portion here. So I have a different additional material used in this class. So complex HLA 1, 2, and 3 region. What is it that? What are the gene products? This 3 is different. We constitutes a complement protein C4 and C2, some tumor necrosis factors, but other product structures we will see that. Remember that here I am not putting like 1, 2, 3 in terms of the classes, because this representation here depicts the representation of this molecule or gene location of these specific gene families in a given chromosome. For example, we will see in the next few slides. HLA system in humans is coded by chromosome 6. And if you take structure of chromosomes in fundamentally, chromosomes ends are called telomeres. Centromere here, chromosomes will have long and short arm. The chromosome 6 short arm at a specific region constitutes a HLA region. And if you zoom in the location of the loci or loci or locus of the each class family's genes, and you look into 5', 3' direction of the transcription. So if you go start with the gene looking from this direction, the first location where the first genes that you can identify on the chromosome are class 2 genes, and the class 1 are located at the end of this HLA region, in between transversed by class 3 genes. And what are those genes? We already discussed class 1, class 2, class 3. And class 1 again, not only A, B, C, they will have again a little bit more broader genes called as E, G, F. And class 2, DP, DQ, DR we spoke, but there are some other families called DM will also exist. They are located in different portions of the chromosome. This is nothing but genetic map of human leukocyte antigen region. If you zoom in, what is the length of the chromosome, length of the region we are looking, HLA region? It's approximately 4,000 kilobases, and of them 2,000 kilobases, major portion will be taken by HLA-1, and HLA-2 is 1,000, HLA-3 is 1,000 kilobases. Again, different class families of the genes are dispersed as suggested in the Kube book. Telomere and centromere using loci position. Different gene families still we are talking. We are not talking about polymorphism set. So, okay, is it clear? So the chromosome, what are HLAs called, where they are located, what are the different genes and different classification of the genes. And one more thing important towards in terms of, we know that there are in a given time, 10^5 molecules of antibodies are present in a given B cell. At the same time, millions and billions of antigen molecules are there. And if you are involving a T cell mediated immune response, then you need multiple molecules. You need the same multiplication, same diversity of MHC molecule that represents attaching to specific T cell kind of interaction. So coming on to these 3 classes, different gene families, how you can produce multiple diverse molecules of MHCs that interact with a specific T cell, how it will happen? It will not happen by the process of somatic recombination. Gene rearrangements will not happen. It will not happen by a process of splice variants production, different mRNAs. It will not happen by that. It will happen by a process called DNA polymorphism. For example, in a given class 2 for the DP, there is one specific locus here. Specific allele will be present given the human type of MHC molecule, but each will have multiple alleles. So we will recap our fundamentals of what are those alleles, what are these polyalleles, then we will come back again. But before that, little bit glance more on the HLA nomenclature in terms of structure and functions. So further in this class, I will be using slides from Professor Lisa Getgender. She is a cancer biologist. She works in Seattle in USA. She is in the Fred Hutchinson Cancer Research Center here. So what are HLAs? What are the molecules in terms of the genes we have seen? in terms of the structure and function, what they will do. Again, different gene location distinguished on the chromosome, class 1, class 2, and class 3, chopped from the chromosome, represented by the different gene classes here, different gene portion here. So what are HLAs? We already spoken. Human leukocyte antigens are the cell surface protein that play a fundamental role in immune response, specifically cell-mediated immune response. Where are they expressed? In terms of which cells they do express? Class 1 MHC molecule that constitute CD8 receptors, they are ubiquitously expressed. In terms of constitutive means they don't need any ligand interaction. They can be expressed on their own on a different cellular molecule. And class 2, they have specific example tissues, for example, T cytotoxic cells, T cytotoxic cells, and they have an inducible expression here. And then in terms of the function, selection of T cell repertoire, initiation of T cell dependent immune response. Okay, class 1 CD8, class 2 4 to the 8, that constitutes CD4 attached T cell receptor. Their main thing, their function is to help the T cell repertoire to elicit immune response. Coming on to in terms of HLA protein structure, whatever we looked in terms of structure of the antibodies, structure of antigens, how the variability will form. That's the same way we looked into fundamentals of primary, secondary, tertiary, and quaternary structure of the proteins coming from their basic amino acid units, alpha and beta helices. Is in the part of the secondary structure. So class 1 molecule, class 2 molecule here. Class 1 molecule is formed from a single chain formed from 2 alpha helices, alpha helices 1 and alpha helices 2, and a beta sheet that will have only one transmembrane domain, and our cytoplasmic tail. You're looking at the plasma membrane here. In terms of the alpha helices, alpha helix 1 comes from a combination of helices alpha 1 and alpha 2. Then alpha 3 independent, they will form a kind of heteropolymer that forms into a portion of this class 1, and specific beta M that is called as a beta sheet formed from a microglobulin molecule. It's a protein 1, protein 2 multi-protein interaction that form one complex of the MHC molecule. And if you look into class 2 protein, it comprised of 2 alpha helices, 2 beta helices, and again coming from 2 transmembrane domains here. And if you look in terms of the location, how they are exposed, the cytoplasmic tails are internal to elicit a specific gene response, but upper to vicinant to the exterior, you see a specific cleft or groove called peptide binding groove that really helps in antigen binding and helps also association with specific T molecules that we will see into that further class. So if you look into the protein structure in terms of class 1, class 2, in terms of the cubae, you can look in terms of the protein fundamental bonds. Same thing, whatever we have spoken in previous slide, constitutes in a different diagrammatic representation. So coming on to the genes, what are the genes that code a specific mRNA and proteins? Class 1, class 2. So the genes in terms of class 1, this is the gene loci, gene locus here, and one of the portion is called A1, A2, alpha 1 and alpha 2. These are the alpha 3 helices, specific location of that. They code specific RNA molecule. This is transmembrane domain portion, mRNA corresponding to that. Proteins will form. They go into a conformational change, protein organization, then go into specific location of the structures. You see here, there is no molecule of beta microglobulin. It may be coded by another gene, but this protein already exists. They may form a complex with that. As such, this gene doesn't code for a molecule of beta microglobulin. Coming on to the molecule MHC2, it will have distinct, again, transmembrane domain. B1, B2, alpha helices are here. A combination of different proteins come, they will form these molecules, not the genes, not the gene variation, gene rearrangement, not the different splice variant. After translation, specific protein molecule complexes will come. One complex in terms of the alpha can combine with alpha into beta. They form a protein molecules in that. So this is again coming from different alpha and alpha 2 as discussed here. Different molecules of the proteins coming from different genes. They go into protein-protein interaction. That's why it's called as MHC complex, multi-intercombinant complex, and molecule coming from protein-protein complexes. So this will represent you in terms of the text required for the understand previous class. And then in terms of the polymorphisms, we are not changing anything here as such. We are not changing any splice variant here, no gene rearrangement here. But still at the end, you are producing different forms of the alpha chain, different forms of the beta chain. Within alpha, you have different polymorphic alpha 1, alpha 2, and alpha 3. How it will happen? It will happen through a process of called polymorphisms in terms of HLA genes, class 1, class 2, where the polymorphism or polyalleles exist. For example, in class 1, you look into the gene. This is a leader sequence that we discussed. It consists of a locus, different alpha 3 and different cytoplasmic tail and transmembrane. But mainly the polymorphism exists in the alpha 1, alpha 2 chain for class 1, and class 2 it exists in the beta 1 chain. So we will see how the polymorphism arise. What are the fundamentally, we'll brush up our fundamentals. So we need to go back a little bit more into Mendelian genetics. We will talk about alleles, loci, homozygous, heterozygous, and little bit on what are haplotypes in terms of looking into HLA typing. So when we talk about genetics, we just talk about father of Mendelian genetics, Gregor John Mendel, who studied the different aspects of genetics of pea plant. And very surprisingly, he doesn't understand about genetic recombination at all. He doesn't know that genes control a specific trait. He was looking for a specific characters that control these, and he defined tall and short, and he know that there is a pattern of heterozygosity, homozygosity, recessive and dominant alleles that we already see looking into the petal color. Homozygous dominant, heterozygous dominant again, recessive, the color changes because this trait is controlled by this allele, this trait is controlled by this allele. And if you think of the alleles, we are talking about specific portion, specific gene that controls the color of the petals, either white or purple. It will have two alleles in the given location, capital B and small b. Capital B is a dominant allele, small b is a recessive allele. It's just an example of biallelic. As humans are diploid, one gets from father, one gets from mother. So we are biallelic in nature. You look in again, and what is a loci? You talk about chromosomes. You talk about B, where is B located? Where is A located? Where is another allele is located? You use physical and genetic mapping. You distinguish them based on that. Coming on to the fundamentals, allele, the DNA sequence at a specific chromosomal location, which presents as a variant, alternative form, tall or short, white or purple. And any given gene will have multiple alleles. So humans, we have two alleles, we are biallelic, but there are polyallelic situation, more than one allele will be present. Biallelic, triallelic, quadriallelic, that's 2, 3, 4. And locus is nothing but a specific chromosomal location, can refer to a specific sequence of a given gene in the DNA. And one more important haplotype. Haplotype is nothing but a set of DNA variations, the polyallelic if presented, polymorphisms at a given locus or given gene location. They might be called as insertions. They might be called it as substitution based on a single nucleotide chains that called single nucleotide polymorphism. They may be insertions and deletions. They are called indels. And remember that these variations in a given chromosome, in a given genetic location, they are present at the same locus, given a gene, given a specific location of the gene. And they are inherited together. When gametes form during reproduction, meiotic crossover takes during cell cycle. But when the polymorphism or DNA variation, they are closely inherited, means they are closely located in terms of the distance between the genes and distance between the alleles. And when the inheritance happens, they go with the crossover segments, they inherit it together. That is one of the fundamental importance. What are SNPs? My variability in terms of nucleotide given location of the gene. I am variable from you. My gene of in given immune response is polymorphic to your gene. That is nothing but single nucleotide polymorphisms. And if you look into haplotypes, given human populations, they may be in a specific location, variable changes will occur. Even the same gene sequence is varied from human to human. and the kind of specific haplotypes can arise given a population. That is importance in HLA type. Coming on to the haplotypes, what are those and how they help in HLA typing and immunology? The terminology changes here. We specifically talk about immune cells. Haplotypes in terms of immunity are closely linked HLA genes coding for MHC molecule that are inherited together are called haplotypes, HLA haplotypes. A genotype in terms of HLA, a combination of parental haplotypes. That's the genotype of specific human being. What is his haplotype for HLA typing? And their codominant expression, they are expressed together in a given locus. And in a given situation, for the haplotype of genotype is always like you don't get same alleles from father, same alleles from mother. You get a kind of cross. So always the genotype is heterozygous for each locus for a specific allele of haplotype. And prevalence is occurrence of antigens between ethnic groups or races, population genetics, linkage disequilibrium, non-random association of HLA alleles. Okay, inheritance again recombination, how the alleles will form. Parents will have two again coming from their parents, maternal. Again, when it goes to recombination, in your case, coming from your parents, recombination happens. See different portions of the color, half and half, different haplotypes can generate by recombination. In the same way, HLA polymorphism, there are multiple alleles for HLAs. I'm not saying about gene rearrangement. I'm not talking about somatic recombination or splicing. I'm just saying that given a form of A class family, there are polymorphic alleles up to this. Given B, two charts, you can see into that, C and D. That's a fundamental process. Multiple HLA types, multiple MHC molecules can be generated. Okay, we have close to 6 minutes. I will try to cover it up, hopefully. Now we will recap again, same concepts. We already brushed up the fundamentals of genetics and polymorphism, fundamentals of immune responses and MHCs and MHC structure, different genes, different molecules and gene segments. So now we're taking this understanding a little bit, go up understanding more of how the MHC system works in immune response. MHC system is nothing but they assist in T cell recognition and subsequent activation, and it's almost fatal to the pathogen, one of the highly potent I discussed. So they will kill virus infected cells, they will elicit macrophages and to kill bacteria, and T helper cells will interact with B cells to activate specific humoral immune response. So this presentation constitutes from Thomas Underhill. It's a very nicely done presentation given the time. I'm following his slides. And how they are developed, how in terms of evolution, it's a kind of a tug of war between host and pathogens, right? Host pathogen responses, immune cells, they evolved. And one of the highly complex and highly stable complex is MHC, and that can kill pathogens. But at the same time, remember that pathogens are also evolving and they are also evading MHC molecule. So there is a co-evolution happening here, a fight. Will you kill me or not? Or I kill you or not? So there is a war between that. And again, fundamental classes we have discussed. MHC class 6 gene families constitute of A, B, C, D, E, F, A, B, C, E, F, and then 5 pairs of alpha and alpha B for class 1, class 2, DP to DO. And they are capable of binding different peptides, approximately 10,000 peptides they can bind. The structure we looked up here. In terms of polymorphism, we are looking into polymorphism here. This polymorphism is nothing but class of the MHCs. We are similar to the antibodies, isotopes and allotopes. Class is there, isotopes are main gene families, and then allotopes are different polymorphism among the families. See into that, in a class A, there are 506 polymorphisms, allotypes are there. Class B, 872, one of the class 2, 466 molecules. They can be constituted in a diagrammatic here, and whatever the color represents, highly polymorphic means multiple alleles, polyalleles are present for each of these specific isotype of MHC molecule. You go into that, class 1, zoom in, they represent CD8 positive T cells, and of these ABC are that E and G interact with NK cells, And you look into the structure here. Class 2, again, these three families CD4, DMDO are intracellular. They regulate peptide loading in terms of attachment here. Look into that. And specifically in class 2, remember that the MHCs are helping antigen processing also, right? You're breaking the antigens into small molecules. So that's why some of these class proteins code for proteasome complexes, where they degrade specific protein molecule, and they help in cytoplasmic to transport ER export transport molecules, and they also assist in peptide loading. For example, a gene TAP is also load for TAP for cytoplasmic ER. So they are associated with antigen processing also. There are more than 100 alleles of a class 1, class 2 loci. Each allele will represent relatively high frequency in a given population. An individual will get the alleles from parents given a chromosome, and it is always heterozygous. It cannot be a case of homozygous, maternal and paternal, and they express together in a given cell, codominant expression. And then how the variability can come up in terms of MHC molecule given the polymorphism, it's nothing but in terms of the amino acid residues. Fundamentally, if you change an amino acid, you change the alpha helix, you change the peptide cleft here. The same way, the polymorphism will produce a change of approximately 20 amino acid changes. Each allele will have a different amino acid combination that gives rise to different peptide cleft. So how it will happen? This is in terms of the molecules of class 1, class 2. The peptide binding motif here, given amino acid sequence N to C. The motifs, for example, class 1, this is a nomenclature we will see in the next class. The peptide binding motif for class 1 will have a differences in location of 2 and 9 monomers, but class 2 will have a heterogeneous population. These are the antigen they are associated with here. Concentrate on this. You read the text here and you go into that. The different amino acids, different residues, changes, variability in the locations of class 1, class 2 constitute for a different combination of anchor residues, peptide binding motif. That single residue change can affect susceptibility and specific resistance. So in summary, there are at least 3 properties of MHC molecule: range of peptides bound, conformation, and interaction with MHC molecule also changes. So we have we came to the end of the class. Almost remember that one more is that MHC restriction. For example, a combination of T cell receptor associated with a haplotype here, bringing a specific antigen here, they will have a ternary complex. They will do the immunoelicit response, but the same T cell recognition cannot go and bind to HLA type 2, type 3, whatever here, different antigen they are bringing. That's called MHC restrictions. This we will talk in terms of xenotransplantation, tissue transplantation. That will be part of the class tomorrow. Okay, hopefully that makes sense in terms of MHCs and in terms of T cell interaction. I will see you tomorrow.